Hi, good morning. Hello, everyone. I'm Marianne from UTAR. I'm also the KLESF Secretariat. I would like to welcome you to today's webinar with title, Could You See the DNA of Living Things? Okay, just a little housekeeping before we get started. If you have any question during the presentation, please type them at the end of the webinar. So I will bring it up during our Q&A session later. Okay, for your info, we are now streaming this webinar live on Kuala Lumpur Engineering Science Fair Facebook and also Minggu Sains Negara Auditorium. So please help us to like and share this video. Okay, and now I would like to introduce our speakers today, Dr. Ong Kiah. All right, Dr. Ong is a senior lecturer at the Department of Science and Engineering, Center for Foundation Studies, Sungai Long Campus, University Tengku Abdul Rahman. As a strong advocate of STEM education, she is actively conducting science workshops for teachers, students, and kids. She has conducted more than 50 sessions of science workshops and hands-on experiment at the public schools and rural area on the voluntary basis since 2014. Her focus is entirely rooted in generating interesting and creative teaching and learning methods that can promote the learning of STEM. Her area of expertise is STEAMs and STEM education. Now, without further ado, I will turn over the time to Dr. Ong. Over to you, Dr. Ong.
Thank you, Marianne. Thanks a lot. And good morning, everyone, and welcome everyone to today's webinar. I'm Dr. Ong from CFS Sungai Long Campus, UTAR. Okay, first of all, I would like to say thank you very much to the organizer for giving me the opportunity to share with all of you on this topic. And it is my pleasure to have all of you in this lovely morning. Okay, so today I would like to share with all of you on this topic, which is known as Could You See the DNA of Living Things? Okay, for this webinar, there are two objectives. First is to explore the simple method to extract DNA from a strawberry. Yes, you are able to extract the DNA at your home, okay, by using the materials and also apparatus that found in your home. And also through this webinar, you'll be learning also the basic principle of DNA extraction. Okay, without further delay, before I start with today's webinar, I have a few questions for all of you. I'm sure all of you have heard about DNA, the term DNA. So what comes to your mind when you hear the word DNA? Think about this. When people ask you what is DNA, so what comes out in your mind? Okay, you can share your idea with me later, okay, after my presentation. You can type it in the chat box so we can have some interactions later on. Okay, so my second question is, what kind of things consist of DNA? So basically, DNA is found in all the living things, okay, but in non-living things, you can't get any DNA. And my third question is, where do living organisms keep their DNA? Which part of the cell that you think the DNA is kept? And my last question to all of you is, in your opinion, what DNA looks to the naked eye? I believe some of you, this thing will come up in your mind, pop up in your mind when people ask you what is DNA and how does it look like, how does to your naked eye. I'm sure all of you, the first thing that will appear in your mind is this double helix structure, right? Okay, but in today's webinar, the procedure, the DNA that you extracted out, right, it is not look like this. Okay, it is not look like this because you have to use X-ray crystallography and so on in order for you to see this double helix structure. And okay, before I move on, right, I would like to introduce all of you to this lady, which is her name is Rosalind Franklin, And this is how Rosalind Franklin looks like. And why is she so, I want to introduce all of you to this lady. This is because she is an expert in X-ray crystallography. And because of her, she produced this picture, which is known as photo 51. This is the X-ray images of the DNA. She produced this photo in 1952. Okay, why is this picture so important? Because this picture, it makes crucial development in the understanding of the DNA structure. Okay, from this picture, from this X-ray picture, we know that DNA is double helix, double-stranded, and it forms helix. Okay, so in one helical turn, the size is 3.4 nanometer, and there are 10 pair of bases in one helical turn. And from this X-ray picture, we also know that the phosphate groups are actually located outside the helix. And the radius for this DNA is 1 nanometer. So she makes crucial development to the understanding of the DNA structure. Okay, so I would like to share with you the second picture. And I'm sure all of you have seen this picture before, right? If I'm not mistaken, you can see this picture when you're browsing the internet, or when you read some articles, or when you read some textbook, biology textbook. I'm sure you will see this two guys picture. Okay, who are they? They are actually James Watson's and Crick. Okay, why they are so famous is because of the DNA double helix structure. Okay, which they developed during 1953. Okay, so this James Watson, Francis Crick, and Maurice Wilkins, they were awarded the Nobel Prize in Medicine in 1962. Okay, from this model, the model that they developed, we know that DNA is made out of two strands, and they are run in anti-parallel. And this double helix is right-handed twisted. And in this DNA, there are base pairing, which is following the Kiah group rules, which means the base adenine is pairing up with the base thymine with two hydrogen bonds, whereas the base cytosine will pair up with base guanine, and there are three hydrogens bond that join both of these bases together. So these two persons are very important in developing the DNA double helix model. Okay, so what is DNA? Actually, DNA is a type of nucleic acid, and it is inherited from the parents. So the parents will pass the DNA to the next generation. And what is the function of this DNA is to direct the synthesis of the protein. Okay, as you can see over here, DNA is a long molecule. Okay, it's a long strand. So it is very difficult for this DNA to pack or to fit into the cell. So it needs some proper packaging, okay, in order to fit well into the cell. That's why this DNA long strand are tightly packed or tightly coiled with this histone, and it will form chromosome. So chromosome is a proper packaging for the DNA in order for this DNA to fit into the nucleus of the cell. Okay, so let's move on and explore what are the things that found in the DNA. Okay, DNA is a macromolecule. That means it is a huge molecule. It is made out of a lot of nucleotide. This nucleotide is the basic unit that make up the DNA. So if you look at the picture of this nucleotide, you will notice that this nucleotide is made up of a phosphate group, a carbon sugar, 5 carbon sugar, and also base. There are 4 types of bases that can be found in the DNA. It can be thymine, adenine, cytosine, and also guanine. So the picture in the center, it shows a single strand of DNA. As you can see from this picture, each of this nucleotide is joining together by phosphodiester bond. Okay, so phosphodiester bond is the chemical bond that join all this nucleotide together to form a polynucleotide. Okay, as you can see over here, the backbone of this DNA is made out of sugar phosphate. That's why it is known as sugar phosphate backbone. Okay, from the model that developed by James Watson and Francis Crick, they show that each DNA is made out of 2 strands. Okay, so you can see over here, it is made out of 2 strands and they are arranged in anti-parallel. That means one of the strand is arranging from 5' to 3', but the other strand is running from 3' to 5', anti-parallel. Okay, 2 strands, anti-parallel. And also from this picture, you can see that the base adenine is pairing up with the base thymine, and there are 2 hydrogens bond that join this, that link these 2 bases together. Whereas for the base cytosine and the base guanine, they are paired up together by 3 hydrogen bonds. So this will be the structure of the DNA that you normally see in some biology textbooks. Okay, is it possible to extract your DNA or extract strawberry DNA at home? The answer is yes. It is possible for you to do that. Okay, by how? Okay, I would like to show you the steps. Okay, so in order to extract the DNA from the strawberries, you need some apparatus and materials. Okay, this will be the things that you need. So you need detergent, you need a glass, you need 70% ethanol, you need strawberries, you need salt, measuring cup, Ziploc bag, a stick or a chopstick, and also ice cubes. Okay, you can easily get this materials and apparatus at home. So after you prepare all these materials and materials and apparatus, let's move on to the steps. Okay, so before you start to extract the DNA from strawberry, you need to prepare buffer, or what we call as extraction buffer. So how to make 50 ml of buffer? First of all, you need to measure 45 ml of water, and you measure 0.75 gram of salt, and you add the salt into the water. After that, you have to add in 5 ml of detergent. And by using a chopstick or a stick, you have to stir well the mixture so that it is well dissolved. And finally, your extraction buffer is ready. After you prepare your extraction buffer, now we can go to the procedure to extract the DNA from strawberry. Okay, so first of all, what you need to do is you need to take your fruits, okay, strawberry, for example, you have to wash it, you have to clean it, you have to remove the leaves. After that, you have to place the strawberry into a Ziploc bag, and you have to seal the Ziploc bag properly, and you can start to crush the strawberry by using your hand. In the third step, you have to open up the Ziploc bag, you pour in the extraction buffer, and you seal tightly the Ziploc bag and leave the mixture for about 2 minutes for the reactions to occur. After 2 minutes, you can use a sieve to help you to separate the pulp and the juice. If you don't have sieve, you can still remove the pulp and the juice by just pouring out the juice from the Ziploc bag, but you have to be more careful. But if you want to make your work easier, you can use a sieve to help you with this. And after that, after you obtain your strawberry juice, you can slowly pour in equal amount of ice cold ethanol into the glass. Okay, as you pour in the ice cold ethanol, you will notice that there are 2 layers of solution formed. The top layer will be the ethanol, and the bottom layer will be the strawberry juice. Okay, you will see that the DNA will start to precipitate and float on the top layer, okay, because DNA is not soluble in the water. So you will see something like this, okay? If you are doing this experiment at home, you will notice that the DNA will precipitate and float on the top layer. Okay, besides using strawberry, you can use different types of fruits as well. Okay, it depends. You can use, you can try with banana, you can try with apple, you can try with pineapple, okay, or other types of fruits, okay. Or if you want to try with vegetables, also can. So you can use the same method, but you change the fruits into other materials. So these are the process or the procedure for you to extract your DNA. Okay, I would like to share a video with you on the whole process. So as you can see from this video, at the end of this video, there are 2 layers of solution form. And if the ethanol is cold enough, you have to make sure the ethanol that you use is really cold. Okay, otherwise your DNA will not precipitate out. So cold ethanol, it will enable the DNA to precipitate. So this is the result that you obtain if you extract the DNA from the strawberry. Okay, so you already learned about the steps on how to produce the buffer and also the steps to carry out in order for you to extract the DNA from the strawberries. So these are the few precautions that you have to take note when you want to extract the DNA. First, you have to make sure the Ziploc bag is tightly sealed. Okay, otherwise all the things in your Ziploc bag will actually come out if you are not sealing it properly. And secondly, it is important for you to not crushing the strawberry for too long. Okay, you only need to crush it for about 2 minutes. Okay, and thirdly, you have to make sure the pulp is properly removed. That's why you can use a sieve to help you in this process. Okay, if the pulp is present, it will actually affect the DNA that precipitate out. Okay, and also you have to make sure the ethanol that you use is ice cold. Okay, so you already learned about the process on how to extract the DNA from strawberry. But how about human being DNA? Is it possible for you to extract your own DNA? I believe some of you will say that yes, it is possible. Some of you maybe you think that maybe it's not possible. You have to go to the lab in order for you to do this. Okay, my answer is yes. Okay, you can extract your own DNA at home. But how? Do you need to cut your finger to draw your blood in order for you to get your DNA? No, no, no. You don't need to hurt yourself or injure yourself or prick your finger to get your blood in order for you to extract your own DNA. So you can extract your DNA by getting your cheek cells from your mouth. Okay, you can extract your own DNA by using cheek cells. So let's check it out on the procedure. So to extract human DNA, first you need to get 45 ml of water and you measure 0.75 gram of salt. You pour into the water and you use a chopstick or a stick to stir well the salt water. Make sure the salt is totally dissolved. After that, you have to vigorously gargle the salt water for 1 minute. After 1 minute, you spit the salt water into a glass and you add in 5 ml of detergent into the glass and stir the salt water and leave it for 2 minutes. After 2 minutes, you add in equal amount of 70% alcohol, and again, you will see 2 layers of solution form. So after some times, you will see that the DNA actually precipitate out. So the process of extracting the DNA from the strawberry and also the DNA from human being is more or less the same. Okay, you're still using 45 ml of water, you're still using 75 gram of 0.75 gram of salt. The only difference is you are not adding in the detergent at the very beginning. Okay, because I'm sure you don't want to gargle the water that contains detergent in your mouth. Okay, so you add in the detergent in step number 4 over here. Okay, so the process is still the same. The materials is still the same, just that the sequence is slightly different. Okay, so after you learn about the procedures, let's learn about what are the principles that behind the procedure, why you do this, why you do that during the experiment, or why are you adding certain types of chemicals, why are you adding salt. Okay, let's learn the basic principle behind the procedures. Okay, what is the purpose of crushing the strawberry? Take a few minutes or few seconds to think about that. Why do we need to crush the strawberry? If you don't want to crush the strawberry by using your finger, you can actually use a blender and you just put in your strawberry into a blender and you start to blend them. Okay, you can either use a Ziploc bag or blender to help you to crush the strawberry. What is the purpose of crushing the strawberry? This is because strawberry is a plant cell, and plant cell consists of cell wall. And I already mentioned earlier on, the DNA is found inside the nucleus, and this nucleus is found inside the cell. So in order to expose the DNA or expose the nucleus to the environment, first of all, you need to break the cell wall. Okay, you need to break the cell wall so that the nucleus can be released to the environment and the DNA can be released later on. Okay, so that is the purpose of crushing the strawberry. And why would you crush the strawberry for 2 minutes instead of 10 minutes? Okay, in one of the procedure and one of the precaution, I already mentioned that you can only, it's good for you to crush the strawberry for about 2 minutes. Okay, you are not encouraged to crush the strawberry for 10 minutes. Why? This is because in the cell, it contains a kind of enzyme which is known as DNase. Okay, what is the function of this enzyme DNase? So DNase is an enzyme that will break down the the long strand of DNA. It looks like a scissors. So if this DNase is active, if the enzymes become active, it will actually cut or cleave the DNA into fragments. So if this DNase is very active, it will cleave the long DNA strand into fragments. So ended up, you are not able to get a very long DNA strands. Okay, you will get the DNA in fragments form. Okay, so when you crush the strawberry for too long, you are actually applying more heat to the DNA enzymes. Okay, when the DNAs get more kinetic energy. Okay, applying heat, it get more energy, it will start to become active. So when it active, it will actively cleave the bonds that found in the DNA. So end up, you are not able to get a long DNA strands, but you will get it in like fragments. Okay, so the result won't be so good. Okay, so what is the function of detergent? If you notice, right, in one of the steps, when you want to prepare the buffer, you need to add in detergent. Okay, what is the purpose of having detergent in the buffer? What is the purpose of having that? Okay, before you can answer this question, I have another question to all of you. Okay, after eating, you always use detergent to wash dishes and clean your hands, right? Okay, why do you use detergent? What is the purpose of having detergent? Take a few seconds to think about this. Why you are not using other materials to clean your dishes and also clean your hands? Why are you using detergent? Okay, so to answer this question, we have to look at the properties of detergent. Detergent has amphipathic properties. That means detergent has hydrophilic properties and hydrophobic properties. That means detergent can dissolve those molecules that are polar and non-polar. Okay, and the plus, just now I already showed you that the DNA is found in the nucleus. Okay, it's found in the nucleus, and the nucleus is bounded or surrounded by a membrane. Okay, so this is how the membranes look like, and this membrane is phospholipid bilayer. Phospholipid bilayer. So it is made up of phospholipid molecule, which means it has hydrophilic head and hydrophobic tail. So if you use detergent, this detergent, it will break apart. Okay, it will break apart the plasma membrane. So when it breaks apart the plasma membrane, the content in the nucleus can be released to the environment. Okay, that's why we are using detergent, okay, to break apart the membrane of the nucleus so that the content in the nucleus, such as DNA, can be exposed to the environment. Then you can precipitate the DNA. Okay, that is about the function of detergent. And lastly, what is the function of salt? Why are we putting salt into the buffer when you want to extract the DNA? Or why you use salt and you dissolve in water and you gargle it in your mouth? What is the function of this salt? Okay, so it serves as two functions. First, you have to look at the structure of nucleotide. Okay, as I mentioned earlier, nucleotide is made up of three components. They are phosphate, 5-carbon sugar or deoxyribose, and also base. So this phosphate group, it is negatively charged. Okay, it is negatively charged. So you need to have this salt to neutralize the negative charge of the phosphate group so that all the DNAs will clump together. That is why we add salt into the buffer. Besides that, there's also another function for the salt. Okay, so as I mentioned earlier, DNA is a very long molecule. So it is very difficult for it to get packed into the cell. So this long strand of the DNA, it has to be coiled together with the histone, and it is proper package, under proper packaging, it will form the chromosomes. So as you can see from this picture, there are a lot of proteins, which is known as histone. So one of the functions for salt is to remove the protein that bound to the DNA, so that ended up you are able to extract slightly pure DNA, okay, the DNA that without the protein. So these are the two functions of salt. First is to neutralize the phosphate group found on the DNA, and secondly, it removes the protein that bound to the DNA. So these are the functions of salt. Okay, so what is the function of ice-cold ethanol? Okay, as you can see over here, throughout the experiment, I'm using ice-cold ethanol. Okay, what is the purpose? Okay, because DNA is insoluble in ethanol, if you use colder ethanol, there will be more DNA that precipitated. So if you want to conduct this experiment at your home, please use colder ethanol in order for you to get best result. Okay, so you have learned about the procedure, you have learned about the principles. Okay, now I have some questions for all of you. Okay, in your opinion, are the DNA of every plant and animal the same? I'm sure some of you will say the DNA in the plant and the DNA in the animal are different, totally different. Okay, some of you maybe you will comment that the DNA is the same, but some of you will say maybe it's different. Okay, why? I give these questions because some of my students, when I was conducting my class, and then some of my students say that the DNA in the plant and the DNA in the animal are totally different. Okay, if you look at the structure of the DNA, which is a double helix that I showed all of you earlier on. Okay, all the DNA in the plants and all the DNAs in the animals, they are having the same structure. Okay, they are made up of two DNA strands. They are anti-parallel. That means one of the strand is running from 5' to 3'. The other strand is running from 3' to 5'. Okay, and okay, and this DNA that pick up the plant and make up the animals, right? They also have the same unit, which is the nucleotide. Okay, it also comprises of phosphate, 5 carbon sugar, and bases. Okay, what makes the DNA of the plant and animal so different is because of the arrangement of the bases. Okay, the arrangement of the bases, the sequence of the bases, and also the number of bases. Okay, for example, maybe a plant, right? The sequence is A, adenine, adenine, thymine, thymine. Maybe another organism is cytosine, 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 cytosine. So the arrangement of the bases that make every one of us or every organism become different. Okay, the arrangement of bases and also the number of bases. Okay, certain organism maybe they have more bases. Okay, the sequence is longer, but some of the organisms they have lesser bases. Okay, shorter DNA. So that's make every one of us become so different and so unique. It is because of arrangement of bases and also the number of bases that found in the cell. Okay, so the DNA structure is still the same, still double helix, right-handed twisted, two DNA strands, anti-parallel. Everything is the same. still consists of phosphodiester bond, hydrogen bond. Okay, so the only difference is the arrangement of bases or the sequence of bases, and also the number of bases that make every one of us become so unique. Okay, my second question, what will happen to the DNA after you consume them? Have you think about this question earlier on? I believe that you eat vegetables, you eat meat, you eat fruits every day in your daily meal. So all these fruits, vegetables, and also animals that you consume, right, consists of DNA. After you consume those DNA in your body, right, what will happen to this DNA from the animals, from the meat, from the fruits? So what will happen to the DNA? Will it become part of your DNA? Think about this. Okay, actually what happened is after you consume this DNA into your body has some enzymes. It will digest the DNA and break it down into a smallest molecule. Okay, then your body will actually recycle all those materials after you consume them. So don't worry, the DNA that you consume, right, will not stay forever in your body, but it will be degraded or it will be digested by your enzymes in the body or by your digestive system. Okay, my third question is, what are the factors that will affect the DNA, the amount of DNA that collected? What are the factors? Okay, some of you, maybe you will say that the amount of fruits that I'm using, if I use more fruits, then I will get more DNA. Okay, that is correct. Okay, first, the amount of samples that you use will affect the DNA that you collected. If you use lesser samples, of course, you will get lesser DNA. Okay, besides that, what are the other factors that will affect the DNA, that amount of DNA that collected? Any one of you have ideas? Okay, you can put in the meeting chat later on, okay, after I finish this session, so you can put your ideas in the meeting chat. And what else? Yeah, I recall something. It's the percentage of ethanol that you use will affect the amount of DNA collected. Yeah, there's one thing that I would like to highlight over here. If you are extracting the DNA at your home, please use at least 70% of alcohol. It's 70% of alcohol in order for you to get good result. Okay, if you use the alcohol that you use, the percentage is slightly lower, you are not able to get very good result. Okay, maybe some of you, you will think that I have some beer at my home. Okay, I have some whiskey, I have some alcoholic drink at my home. Can I use those alcoholic drink to do this experiment to extract the DNA? The answer is yes, you can use the ethanol or alcoholic drink to extract the DNA, but it is very difficult for you to get good result. Why? This is because those beer, alcoholic drink, the content or the percentage of ethanol is very low. Okay, it is very low. So the DNA are not going to precipitate. So you must use at least 70% of alcohol in order for you to get good result. Okay, of course, if the percentage of alcohol higher than 70% is better. Okay, you can try with 75% of alcohol, maybe 95% of alcohol. Okay, the higher the percentage, the better. Okay, so the DNA collected will be affected by the percentage of alcohol that you use. Okay, you have to use at least 70% of alcohol. Okay, you can get this 70% of alcohol from any pharmacies. And also, what are the factors? Okay, besides amount of samples, besides the percentage of alcohol. Yeah, another thing that I keep on emphasizing just now is the condition of the alcohol. The colder, the better. Okay, I have tried earlier on. If I use normal room temperature, 70% of alcohol, I'm not able to get very good results. There are only very little amount of DNA that precipitate as the result. Okay, so you must make sure when you are doing this experiment at your home, you want to collect the DNA at house. So please make sure that the alcohol that you use is cold enough. Okay, it's ice cold, ice cold. So besides amount of samples, beside the percentage of alcohol, beside the condition or the coldness of the alcohol, what are the factors that also will affect the DNA, amount of DNA that collected? Any one of you can think of other factors? Okay, maybe some of you will give the answer. If you crush the samples or crush the strawberry for too long, and then you are not able to get a very good result, right? Just now I already mentioned that the DNA's enzyme become very active and it will cleave the DNA into fragments. So it will affect the DNA of the amount of DNA collected. Okay, that is a good factor also. But there's one more factor that I would like to share with all of you. Okay, is this thing, Ploidy. Okay, Ploidy number, it means the number of sets of chromosomes in a cell. Okay, I'm sure most of you are quite familiar with haploid, diploid, right? You learned during your biology class, haploid, diploid. Okay, haploid means there's only one set of chromosome. Diploid means there are two sets of chromosome. Okay, we as a human being, the cells in our body, all the cells in the somatic cells in the body is 2N, but not all the cells in our body are 2N. Some of them are haploid. Okay, for example, your gamete, okay, N, haploid. Okay, if there is only one set of chromosome, we call this as haploid. If two sets, we call this as diploid. 3 sets, triploid. 4 sets, tetraploid. 5 sets, penta. 6 sets of chromosome, we will call this as hexaploid. 7 sets, heptaploid. 8 sets, octaploid. So the amount of DNA that you collected, right, is actually affected by this Ploidy number. If the Ploidy number is higher, you will get more DNA. Okay, the DNA that precipitated will be more. If the Ploidy number is very low, the DNA that you can see or the DNA that you can collect throughout the process, right, is very low. For example, if you are extracting the DNA from strawberry, you can see a lot. But if you want to extract your own DNA, human DNA, the amount will be very little. Why? Because your chick cell has 2 sets of chromosomes only, which is diploid. So you will see only very little of DNA that precipitated, okay, as the result. Okay, so if you are not very clear, I will show you this picture. Okay, this picture, it shows you haploid, diploid. Okay, as you can see over here, haploid means there's only one set of chromosome. Diploid, so each copy there are 2 sets, okay, of chromosome. So triploid, tetraploid, and hexaploid. Okay, so as I mentioned earlier, some plants they have higher Ploidy number. Okay, for example, strawberry is octaploid. Okay, octaploid means it has 8 sets of chromosome. For wheat and sweet potato, it has 6. Both of these plants have 6 sets of chromosome, or what we call as hexaploid. For blackberries and also peanuts, there are 4 sets of chromosome and they are known as tetraploid. As for watermelon and banana, these fruits contain 3 sets of chromosomes which are known as triploid. Okay, so from this few examples, right, you will notice that strawberry has higher Ploidy number, okay, 8N. Okay, if you extract the DNA from the strawberry and you extract the DNA from the banana together, okay, let's say you are doing this experiment at home together, you have strawberry and banana with you, and you extract the DNA from strawberry and banana together, okay, you are using the same amount of samples. Let's say you are using 10 gram, you are using 10 gram of strawberry, at the same time you are using 10 gram of banana. Okay, if you compare the result, you will notice that strawberry, you will get more DNA. But for banana, you will get lesser DNA, that precipitate. Why? This is because of the Ploidy number. Okay, strawberry has 8 sets of chromosomes, but banana has only 3 sets of chromosomes. So even though you are using the same amount of samples, but the DNA that you obtain will be totally different. Okay, because of the number, the Ploidy number. Okay, you can try this experiment at home. So by extracting DNA from different samples. Okay, so that is the end of my presentation. And thank you, everyone. Thanks for attention. And if you have any questions, please feel free to email me through this email address. Thank you, everyone. Do you have any questions?
Okay, thank you, Dr. Ong, for the sharing of your knowledge. It is definitely very useful information for our students. So now we will go ahead and take some time for questions. So if you have any questions, you can post your question in the chat box. We will also check if there's any question posted from the Facebook. Dr. Ong, I believe you could also see the chat box, right?
Wait, let me check. Is there any questions from the floor?
So far, okay. There is one posted by Nadra Harith. The question is, can we use other parts of the plants, such as leaf or stem?
Yes, can. You can use different parts of the plants when you want to extract your DNA. If the plants or the samples that you are using is slightly harder, right? It's very hard for you to break the cells. You can use a blender to assist you. Don't worry. Okay, because the blender it can help you to break the cells also. But again, as mentioned earlier, please do not blend your samples for too long, because when you blend it for too long, so the DNA's enzymes will get activated, so it will actually cleave the DNA that found in the plant. So ended up you are not able to get a very good result. Okay, you will get like fragments, a lot of fragments.
Right. Another question here from Golding Yuan. May I ask what is inside the nucleus and the molecule?
Okay, in the nucleus, of course, you can see a nucleoplasm, okay, which is the cytoplasms that found in the nucleus. And also you can see some rRNA or ribosomal RNA. You will see nucleolus also, and you will see DNA. Okay.
Dr. Ong, I think your mic is muted.
Yeah.
Yeah, okay, sorry.
Where should I start? Okay, what are the things that found in the nucleus, right?
Yes.
Okay, so basically in the nucleus, you will see nucleolus, okay? You will see rRNA, you will see DNA, and you will see some proteins. Okay, so these are the things that found inside the nucleus. Okay, what else?
Okay, the next question will be from Tai and Kiah.
Water have DNA?
Yes.
No, because water is not a living organism.
Okay.
Whether greater.
All right, another one is from Ang Yi Jin. Good day, Dr. Ong. May I know whether greater Ploidy, Ploidy numbers indicates greater storage of genetic information for the particular species?
I can only say that when the Ploidy number is higher, there are more DNA sequence. Okay, but I'm not very sure that whether it indicates greater storage. Okay, because number of bases is not equivalent to greater storage. Okay, I can only say that there are more DNAs in the cell.
Okay. Next question from Tanjun Chen. Dr. Ong, I read from an article DNA is fireproof. What is your opinion about this?
Wow, this is a very good and interesting article. Is it fireproof? I need some time to think about this question. Is it possible for me to email you and answer your questions later on? Would you mind to share with me the article?
Sure. So maybe later,
Yeah.
you can post the question in our KLESF Facebook. We will get Dr. Ong to reply you there. All
Yes.
right. Okay. Dr. Ong, another one from Go Bing Yuan.
Okay.
I think he answered that water doesn't have DNA. What is cytoplasm? Is it?
Cytoplasm is something like liquid things that found in the cell. Okay, it's like gel form to allow some chemicals reaction or biochemicals reactions that occur in the cell. Please clear up again.
Okay, there's another question from Padmavati. Please clear up regarding Japanese scientists once told that water has memory. Please explain. Okay, Dr. Ong,
that is very interesting, and this is very new to me. I think before I can answer this question, I think I need to do some read out and some research on this. Then I hope I can reply your questions as soon as possible. So I need some time to do some research on this before I can comment on this statement.
Okay, probably Padmavati could also post the same question in our KLESF Facebook. We'll get Dr. Ong to revert back to you. All right. Next one is from Ang Yi Jin. Okay, the question is, Doctor, is having more DNA better for survival?
I think it is possible. It is possible, but I'm not so sure whether if there are more DNA, we can have better survival. Okay. I can maybe if you have more DNA, that means you have more sequence bases, right? That means maybe you can produce more protein, and maybe it can help for better survival. I also need to do a research on this before I can really confirm the answer. The chlorine can destroy human DNA. Is that true? Yes, chlorine is a kind of chemicals, and it can actually degrade the DNA. Yes, human DNA. Where does materials comes from?
Yeah, there's another question, Dr. Ong, from Go Bing Yuan. Where does the protein and bacteria come from?
You mean the protein in your body? Are you trying to mean that? The protein in your body is actually encoded in your DNA. So your DNA, right, is made up of a lot of bases, and this bases, right, will be translated into protein, and then this protein will be used in your body. Okay, so the protein is actually coming from the DNA that code, from the sequence of DNA that sequence of bases that code in your DNA. Bacteria comes from bacteria's. That will be a very long story because it involves evolutions and everything. So I think the time is not enough for me to explain about where the bacteria comes from, because due to the time constraint. If you maybe you can leave your email address, and then I can share with you some articles on where bacteria comes from.
All right. Okay. So let me see if there's any question posted in Facebook before we can proceed. Okay, there's no other question from the Facebook, but okay, there is a question here in our chat box. Ang Yijin, the question is, Doctor, could you please explain how we can get rid of DNA from certain area, example floor, table, since DNA is easily found everywhere?
DNA is easily found everywhere. Basically, DNA is found inside the cell. So you need to, it is not like spreading everywhere. You can't really see DNA double helix everywhere because they are actually well protected in the nucleus or well protected in the cell. So it is not, you can't get DNA everywhere unless there is a cell, okay? Because DNA is found inside the cell. So if you clean up the places is without the cells, without the bacteria and everything, so you won't be having DNA everywhere. How do we rewrite DNA? Rewrite DNA, right? It needs a lot of studies. That means you need to really know the sequence. Okay, it involves a lot of processes for rewriting DNA, and it is not an easy thing to do. It takes time, it takes years to really study the sequence of the DNA before you can rewrite the DNA.
All right, thank you, Dr. Ong. Oh, I believe Eugene is posted another.
The synthetic rubber like tire contain DNA? The answer is no. The answer is no.
Okay. So, Dr. Ong,
yes?
There is no other question posted in the Facebook. Sorry, it's from, okay, but in our chat box, you can see Dayanita.
Okay, what is meant by genetic modification? Okay, genetic modifications, right? It meant you go and change the sequence of the genetic. Okay, for example, initially, right, the sequence I showed you earlier on, the DNA, it is a long polynucleotide chain, so there are a lot of bases. Okay, so for example, now the sequence is AATTT. So if you modify them, maybe it becomes GGCCC. So that is the meaning of genetic modification. You go and modify the sequence, or you go and modify the bases that found in the polynucleotide chain.
Okay. So hopefully it's.
I hope I.
Okay. Yes.
Yes.
All right. Okay. Okay, Dr. Ong.
Yes.
I think if you don't mind, can you share again your email address from your slide?
Sure.
So for our participant, if you have any other question after this, feel free to send it to Dr. Ong via her email, or you can also post your question in our Facebook, and we will forward it to Dr. Ong for her reply. Okay, so there's no other questions from our Facebook and also MSN YouTube channels. Okay, before we ended the session, okay, I would like to tell our participant that We have prepared a feedback form in Facebook. We already posted it in our Facebook, and we will also post it in our Zoom chat box. So for participants, please kindly fill up the feedback form after the webinar, because your response are very important in helping us to assess the effectiveness of the webinar. So we appreciate you for being here. Before we end this session, Dr. Ong and the participant, if you don't mind, we like to take a screenshot of group photo. So everyone in the Zoom, please turn on your camera. Okay, has everyone
Dr. Ong, you need to stop sharing the slide.
yes. Okay, so everyone, please be ready. We're going to take a screenshot in a short while. Give me one moment. Okay, if you're ready, put on a smile on count of 1, 2, 3. Okay, that is the first shot. Okay, we will take another one. Okay, 1, 2, 3, smile. All right. Okay, thank you everyone. Thank you Dr. Ong for your time today. Okay, before we ended the session, I would like to inform our participant that for the next event, we will also have another webinar at 11:30. The title of the webinar will be Video Editing for Beginners. The speaker will be Miss Cindy Yoh Lee Cheng. She is one of UTAR lecturer. So if you are free, please join us. The Zoom link will be the same. And you can also watch the next webinar in our Kuala Lumpur Engineering Science Fair Facebook channel. So Dr. Ong, if you have any other message for your participant today.
Thanks everyone. Thanks for attending this webinar. I hope you learned something, and yeah, it's good to it's nice to share with all of you. Thank you everyone.
Okay, thank you Dr. Ong for the sharing today. So thank you everyone for your time. So hope to see you later. Okay,
Bye,
thank you.
everyone.
Okay, bye.
Bye.